The primary objective of the translational research centers in thrombotic and hemostatic disorders, also known as TRC-THD, is to enhance the translation of basic research discoveries that would lead to improved prevention, diagnosis, and treatment. The TRC-THD program brings together clinical, translational, and basic investigators for a highly interactive, multidisciplinary environment to address important clinical questions. The research focus is on early translation of basic research discoveries, testing their application, feasibility, and the possible clinical benefit to society. A translational research skills development component provides a mentored research experience to young investigators who acquire the competencies needed for translational and clinical research studies. To enhance the potential success of this new program, the centers were required to leverage existing infrastructures and resources at their respective institutions through collaborations with their CTSAs, or comparative institutional programs. This would enable a timely and cost-effective completion of proposed translational and clinical research projects. Expected outcomes from this program are the identification of predictive markers of risk, the development of improved diagnostic tools, and the discovery of new therapeutic agents. Let me tell you a little bit about the consortium. It's made up of 5 research centers and an administrative coordinating center. The consortium is overseen by staff from the National Heart, Lung, Blood Institute, an external advisory committee made up of scientists from non-participating institutions, and a steering committee consisting of principal investigators from each research center. The consortium also has a skills development core subcommittee to synergize the educational experience and opportunities within the research centers. Over the next several minutes, you will be introduced to each of the center principal investigators who, in turn, will summarize the work being performed at their respective centers. But I would first like to introduce Dr. Rebecca Link, Program Director from the National Heart, Lung, Blood Institute. Tell us about this program.
The Translational Research Centers in Thrombotic and Hemostatic Disorders was developed to enhance translation of basic research findings toward clinical application. The emphasis is on early translation, on testing the applicability and the feasibility of basic findings toward the clinical application. Some criteria that was asked for each of the centers who applied was that they would have components for both translational research, but also for a discovery component and a clinical component, so that there was the full spectrum and investigators would see and interact across the full spectrum from discovery to clinical application. Another component that every center was asked to have was to have a career development component, so that young investigators would be exposed to this experience and to learn about translational research. We also asked the centers to leverage the capabilities and resources at their institutions and apply them to this program. Currently, the program is about halfway. It's about 2.5 years into a 5-year program, and at this point I would say the centers, all the centers, are exceeding my expectations. What I have found is, and I would owe that to the strength of the investigators. All of the investigators are outstanding, and they had created outstanding teams. They not only have good collaborations within their individual centers, but we have seen collaboration across centers. I would like to give credit also to the Duke Coordinating Center, because I think they have really helped this interaction so that we see a strength I think having the focus and everybody's interest on thrombotic and hemostatic disorders has also really helped the program, so that there is a mutual interest and understanding in moving this area forward.
Good. Thank you, Dr. Link.
Thank you.
And Bruce, tell us a little bit about the work being done at Duke University Medical Center.
Well, thanks, Rick. It's a pleasure to tell you about the work we've been doing in conjunction with the U54 program from the National Heart, Lung, and Blood Institute, focused on translational thrombosis type of studies. And what we've really focused on is trying to figure out how to make more effective and safer therapies targeting part of pathological blood clots called platelets. And so our work is really focused on how do we make novel antiplatelet agents, but ones that have a safety switch where we can turn them off. And as you know, in many sort of acute care settings where we're doing surgery or angioplasty or putting a stent in a patient, one of the challenges is we don't want people to clot during the procedure, but then because we've either done surgery on them and cut into them or put a catheter into them, and that you can have the propensity to bleed from those access sites. And so one of the things that our group is really focusing on is how do we block that clotting by targeting these important pieces of the clot called platelets, blocking platelet adhesion during the procedure, but then following them, how do we reverse that drug's effect quickly so the physician can turn off that inhibition so that you don't have bleeding complications following those procedures. So with support from the U54 program and the National Heart, Lung, and Blood Institute, I think we're making great strides to develop new agents to do that. So within the program, we're making them, we're characterizing those type of compounds, we're developing ways to monitor them, and then we're translating them into first-in-human studies. Then finally mention the last thing that I think is really important we're doing is we're training additional young investigators who are interested in this area in the long haul to help create the next generation of innovators and translational researchers who are interested in controlling thrombosis and improving treatment for people who have thrombotic disease.
Very, very important, and thank you, Bruce, for joining us today.
Thank you, Rick.
Dr. Wayrick, tell us a little bit about the work that's being done at the University of Utah.
Yeah, so patients with diabetes type 1 or 2 or with obesity are at increased risk for thrombosis. And so at the University of Utah, we're studying the central theme that the metabolic milieu, changes in the systemic circulation like increased glucose, free fatty acids, or things like that with systemic inflammation, can actually give you molecular changes in both the megakaryocyte, which is a platelet precursor, and the platelet. And these changes phenotypically induce a response that makes the platelet kind of hyperreactive, prone to clot. And so we have actually 4 main projects that study that, 2 which are basic science projects, 2 which are clinical projects, and then cores that support that. In brief, the basic science projects study gene expression, how the systemic metabolic milieu changes the gene signature in both the megakaryocyte and the platelet to make them prone to thrombose. That's one that I direct. The second one is kind of how the metabolic changes actually occur. That's directed by Dale Abel, and that specifically looks at glucose transporters and mitochondrial proteins present both in the megakaryocyte and the platelet. The clinical projects, Project 3 is led by Matt Rondina and Guy Zimmerman, and it actually looks at patients that are pre-diabetic and to examine the molecular changes in the platelet that make that prone to thrombose. And then also looking at a drug that's been used, metformin, both pre and post metformin, to see what the molecular changes are. And then the 4th project is with Dr. Jane Friedman up at UMass, and she's done some really great work looking at lipid infusion, looking at the acute effects of lipid, bariatric surgery, so pre and post weight loss, and then actually looking at the Framingham study, patients within that subset that are diabetic or obese, and their increased risk for these molecular expression changes in platelets. So you can see that there's a theme around both the basic and the clinical work. And then we have 4 cores, you know, and one is the mouse and cell isolation, one is kind of for the patients. But the last core, which I think is the most important, is our translational skills development. And within that, we have a group of physician scientists and then some young PhD graduate students who were really preparing them for work both in the obesity and diabetic world and how they can actually interface their science and see how that they can begin to develop careers to look at the roles of platelets in thrombosis.
That is fantastic, and a broad spectrum of science and application. So thanks so much for joining us.
You're welcome.
Dr. Fury, tell us a little bit about the work that's being done at the Beth Israel Deaconess Medical Center, Harvard Medical School.
We are working on a protein called protein disulfide isomerase. This particular enzyme has been well known for a long time and to be important for making proteins inside cells. We discovered as part of our basic research program that this protein actually is secreted by cells during thrombus formation and is required for thrombus formation. Because it is required for thrombus formation, we have hypothesized that the inhibition of this protein could lead to a completely novel approach to developing a new antithrombotic drug, a new drug to treat stroke, to treat pulmonary embolism, to treat deep venous thrombosis, myocardial infarction, etc. With this in mind, our project is focused on identifying protein disulfide isomerase as a target for drug development. And as part of this, we're doing studies in animals as well as studies in humans. Through our initial screens, we've identified some compounds that are actually in general use by the public, such that we can avoid many of the complexities of going through clinical trials.
Fascinating, something that certainly could be applicable, and the cost of development could be relatively modest by current terms.
Perhaps. Again, we don't know whether we're actually going to develop a drug per se, or whether we're going to point in the direction of making an even better drug, which will come as a second or third generation from this work.
Yes, fascinating. Thank you, Dr. Fury. I'm joined by Dr. Pete Lawler from Emory University. Dr. Lawler, tell us a little bit about the work that's going on at your place.
Our project is to work on the biological variation of a disease called hemophilia. And it's a bleeding disorder that's due to the deficiency of a blood clotting factor in blood. And currently, hemophilia is treated with infusions of factor VIII intravenously. And that treatment is not optimal because for 2 reasons. One is it's expensive. 2, it is an intravenous infusion, which is inconvenient. And then there's another complication, and that is some patients that are treated with factor VIII develop an immune reaction to it, or an allergic reaction to it, if you will. And when that happens, then the treatment's no longer effective. So our program is designed to address all of these issues. Project 1, which I direct, it deals with trying to understand how this immune response develops, and we're trying to design a new kind of factor VIII molecule that would potentially get around the immune response, or even once the immune response has started, to try to defeat the immune response. And Project 3 deals with trying to develop some new diagnostic techniques and perhaps a new kind of therapy for hemophilia based on, again, trying to evade this immune response. Project 2 is directed towards trying to develop a genetic therapy for hemophilia in which the patients that have a defective gene receive the factor VIII gene. And that would get around having to have intravenous infusions. However, the work that's been done in gene therapy so far, there is an immune reaction that's involved. So the Project 2, one of the goals of Project 2 is to better understand the immune response that occurs there. And then finally, the fourth project deals with the fact that patients with hemophilia vary in their bleeding problem. Some patients with hemophilia bleed more than others, and we don't understand why. One of the hypotheses is that patients have variation in their platelet function. Platelets are a cell that's involved in blood coagulation, and differences in platelet function could potentially compensate for the hemophilia defect. So Project 2 is directed by Trent Spencer and Chris Doring in the gene therapy project. Project 3 is directed by Shannon Meeks. And then Project 4, which is the biological variation that is focusing on platelets, is directed by Sean Jobe and Amy Dunn. We also have in Project 4, trying to understand this biologic variation, a new technology in which we're creating a device which measures bleeding in an in vitro assay that occurs under conditions that we think mimic what happens inside the body. And that project is directed by Wilbur Lamb.
Fascinating. So a broad-scale approach to a very common disorder.
That's correct, yeah. And the whole project is focused on hemophilia and trying to improve diagnostic and therapeutic procedures in the field.
Fascinating. Thank you so much, Dr. Lawler.
Great, thank you.
I'm joined by Dr. Evan Sadler from Washington University School of Medicine. Dr. Sadler, tell us a little bit about the work that is going on at your place.
Well, we are investigating the role of proteases in the pathogenesis and treatment of thrombosis. The premise is that the major proteases of blood clotting are responsible for the production of blood clots that are important for hemostasis, but when this is not controlled adequately, they cause thrombi that can cause downstream ischemia, infarction, and death of tissue. So we are focusing on microvascular thrombosis and large vessel thrombosis, and have projects that address the role of proteases in causing these different kinds of thrombosis in vessels of different sizes. So we have, for example, two of us are focusing on microvascular thrombosis. Myself, thrombotic thrombocytopenic purpura, which is usually a disorder caused by defects in the cleavage of von Willebrand factor by a metalloprotease called ADAMTS13. And John Atkinson, the director of Project 2, studies the role of defects in complement as they pertain to microvascular thrombosis in hemolytic uremic syndrome, in macular degeneration, in preeclampsia, and some other disorders that are characterized by microvascular thrombosis. And then we have a couple of investigators working on large vessel thrombosis. For example, Enrico De Cera, who's at St. Louis University, a few blocks away from us, he's a biochemist and enzymologist who works on thrombin and has discovered how to engineer thrombin into a protease that does not cleave fibrinogen very well or activate platelets, but is an anticoagulant molecule. So he's basically turning this quintessential procoagulant enzyme into an anticoagulant. And George Brose, working at the initiation phase of blood clotting, as opposed to Enrico, who's working at the final protease. George works on tissue factor pathway inhibitor, which regulates the initiation of clotting. And you could say that both George and Enrico are looking at using blood clotting to manipulate cell signaling as an anti-inflammatory strategy, an anticoagulant strategy, which is very interesting. And finally, we have Sam Wickline, who's a cardiologist who is developing nanoparticles as a technology for imaging, for diagnosis of thrombosis, but also as a mechanism for drug delivery. So I think in this way, through strategies addressing microvascular and macrovascular thrombosis and imaging and drug delivery to thrombi, this comprehensive approach will facilitate the translation of our basic research into the clinic and into the hospital.
Fascinating. Thank you, Dr. Sadler.
Thanks.
Thrombotic and hemostatic disorders in humans collectively represent a major cause of morbidity, mortality, and healthcare expenditures in the United States. The Translational Research Centers for Thrombotic and Hemostatic Disorders is a consortium of experienced research centers dedicated to developing novel approaches to diagnosis, management, and the treatment of thrombotic and hemostatic conditions and disorders. It is a strong foundation of support for scientists and clinician investigators who are focused on the translation of fundamental discoveries to care delivery and potentially to global health.